You know that moment in calculus class where you stare at an integral sign and think, seriously, finding the area under a curve, when am I ever going to use this in the real world?
Oh, absolutely. It feels completely abstract, doesn't it? Volumes of revolution, rates of change.
Yeah.
Just seems so disconnected from anything, well, tangible.
Totally. And specifically, how does any of that possibly connect to something as wonderfully messy and alive as biology?
Yeah. That's $1 million question, right?
Well, it turns out that seemingly abstract math, the ideas behind integrals and differentiation aren't just useful in understanding life. They are absolutely essential to grasping its incredible complexity.
That's exactly the powerful insight we're pulling from the source material for this deep dive today. We've got two main pieces we're looking at.
Okay.
One that traces the, well, surprisingly ancient roots of the integral concept, and another that plunges right into the world of mathematical modeling, particularly in what's called systems biology.
Right, systems of biology. So our mission then is to unpack these sources, find those vital connections, and really reveal how these mathematical tools become the language we actually need to understand living systems.
Exactly. Get ready, because the link between ancient geometry and, modern biology is pretty fascinating.
I'm intrigued. Where do we start with the history?
Let's start there, yeah. One of the sources does a great job looking at where this whole idea of integration even came from. And you might be surprised, it goes way, way back.
Way back, like before Newton and Leibniz.
Yeah.
Before the formal calculus we learned.
Oh, centuries before. The source takes us all the way back to ancient Greece. It mentions figures like Eudoxus and Democritus around 370 BC.
Wow. Okay, so what were they doing?
They were grappling with a fundamental problem. How do you find the area or volume of irregular shapes?
Okay, yeah, squares and circles are one thing, but what about like weird curves or spheres?
Precisely, and they developed something called the method of exhaustion. The core idea is just brilliant in its simplicity, and honestly, it feels incredibly modern in its spirit.
Method of exhaustion. Sounds intense. Tell me about it.
Well, the intensity is maybe in the concept. Imagine trying to find the area of, say, a circle. You could try filling it up with smaller and smaller squares or other polygons inside it.
Okay.
You can easily calculate the area of each of those small, simple pieces.
Right.
And you sum them up. And as you make the pieces smaller and smaller, using an infinite number of them.
You eventually fill the whole thing up. You exhaust the area.
That's the idea. You get closer and closer and closer to the true value. It's about breaking down something complex into simple, measurable bits.
Ah, okay. So it's like approximation, but taken to the absolute limit.
Exactly. And the source really highlights that Archimedes in the 3rd century BC took this method and just ran with it. He used it to calculate things that were previously impossible.
Like what?
Things like the area of a circle, the surface area and volume of a sphere, even the area under a parabola, or the volumes of shapes you get by rotating curves like paraboloids.
That's amazing.
Yeah.
So that core idea, summing up tiny bits to get a total, was there millennia ago.
It absolutely was.
Yeah.
And that fundamental concept is what eventually evolved into the modern integral we see in calculus textbooks.
Yeah.
You're still calculating a total quantity, area, volume, whatever, by summing up an infinite number of infinitesimal pieces.
Right, like when you approximate the area under a curve by drawing those little rectangles underneath it.
Mm-hmm.
And then you imagine making those rectangles thinner and thinner and thinner.
Exactly right. One of our sources uses that exact example, finding the area under the curve of, say, the square root of x between 0 and 1. You can get a rough answer with a few fat rectangles.
Yeah, a very rough one.
But as you make those rectangles incredibly narrow, like infinitely narrow, their total sum perfectly matches the true area. It's the precise sum of all those tiny contributions.
Okay, makes sense. And I guess this idea isn't just stuck on calculating simple areas on a flat page, right? The source mentioned generalizing this.
Yeah, it did. Things like line integrals or surface integrals.
Okay, what are those?
So instead of just summing contributions along a straight line, like the x-axis, you might sum up something along a winding path through space that's a line integral.
Ah, okay.
Or you might sum contributions over a whole curved surface in 3D, a surface integral. The source notes how crucial these are in physics for calculating things like the work done by a force along some path, or maybe the flow of fluid across a membrane or surface.
Right. Okay, so we have this powerful mathematical machinery built on this ancient idea of breaking down complexity to find totals. But the big question remains, How do we bridge this to the incredibly complex, dynamic world inside a living cell?
Okay, that's where the second source, the one focused on systems biology, completely changes the picture. It points out that for a long time, molecular biology was very, well, reductionist.
Reductionist, meaning
You'd study one gene, one protein, maybe one pathway at a time. You'd try to understand each piece in isolation.
which is important work, obviously.
Oh, vital work. Absolutely. But it's a bit like trying to understand how, say, a transistor radio works by just studying a single transistor or maybe a resistor in isolation.
Right. You can describe the component perfectly, but you have no idea how it actually functions as part of the whole circuit.
Precisely. One of the sources uses that exact analogy. It says you completely fail to figure out the radio's function just by describing its parts qualitatively.
Okay.
And biological systems like how a cell processes a signal, or how a metabolic network functions, or how genes turn each other on and off, these are vast interconnected networks.
And their behavior is often weird, counterintuitive.
Exactly. It's often non-intuitive. Just knowing what each individual piece does isn't nearly enough to predict what the whole system will do when it's all interacting together.
So you need something more than just describing the parts list. You need a way to capture the interactions, the dynamics over time.
Yes. And this is where mathematical modeling, as described in this source, becomes indispensable. These dynamic mathematical models are essentially quantitative frameworks. They represent these biological networks and their interactions using the language of mathematics, usually equations.
And these models, they aren't trying to be perfect replicas of reality, are they? They're more like abstractions designed to focus on specific questions.
That's a key point, yes.
Yeah.
You build them to explore a particular aspect or test a specific hypothesis. And then the source talks about this really neat idea, the virtuous cycle.
Virtuous cycle.
Yeah. You formulate the model, that's your quantitative hypothesis, basically. Then you use a computer to simulate its behavior over time, see what it predicts.
Okay.
Then you compare those predictions to what you actually observe in real biological experiments.
And if they don't match up,
Then your hypothesis or your model needs work.
exactly. That mismatch forces you to refine your biological understanding and refine the model itself. It's this continuous loop of hypothesis, prediction, experimental testing, refinement, all powered by math.
Okay, I see the loop. So it's a way to rigorously test our understanding. But where does the calculus, the integrals and derivatives come back in? All right, here's the direct connection. Because the language of these dynamic models in biology is very often differential equations.
Ah, differential equations.
Yeah.
Okay, reminding me here. That's all about rates of change, right? How fast things are changing.
It is. If you want to describe, say, how the concentration of a molecule changes over time as it's being produced and consumed in various chemical reactions.
Or how the voltage across a nerve cell membrane changes as ions flow in and out.
Exactly. You use a differential equation. It tells you the speed and the direction of change at any given moment based on the current state of the system.
Okay, and the integral, that's the inverse of the derivative, right? So if the differential equation tells you the rate of change,
The integral lets you figure out the total change or the accumulated amount over some period of
goodness, a
time.
link. Okay, so the models use differential equations to describe the instantaneous rates. And then understanding the overall behavior, finding the actual levels or amounts at different points in time, often involves solving or analyzing those equations, which fundamentally relies on calculus concepts like integration and differentiation.
You got it. One of the sources even gives these classic physics examples, but points out how they apply right here in the biological modeling context.
Like the one about velocity and position. If you know a particle's velocity, its rate of change of position.
Yeah, velocity.
You integrate that velocity over time to find its total displacement, like how far it moved between time A and time B.
Exactly. You're going from the rate velocity back to the total accumulated change. Displacement xb minus xa. That's the integral from A to B of vt dt.
Or calculating work done by a force, if the force changes depending on position.
Link effects.
You integrate that force over the distance to find the total work accumulated as you move from A to B.
Yes, the integral of fx dx. These really show how integration is the tool to find the total effect, or the final state, given the rate at which something is happening.
And this whole quantitative approach, using these mathematical tools, is applied to understand just incredibly diverse biological systems. The sources list a whole bunch.
Oh yeah, we're talking about the real nuts and bolts of biochemistry. Things like biochemical kinetics, the rates of enzyme reactions, how inhibitors slow them down, complex stuff like allosteric regulation, where molecules bind away from the active site.
Or mapping out entire metabolic networks, figuring out how the flow of molecules through pathways is controlled. Like that classic example of end product inhibition, where the final product of a pathway slows down an early step.
Right, a feedback loop. And signal transduction pathways are a huge area too. How do cells receive signals from outside and process them internally?
Like how bacteria sense chemicals.
Yeah, bacterial two-component systems, or in our cells, G-protein coupled receptor pathways, chemotaxis, how cells sense chemical gradients and move towards or away from them.
The sources mentioned calcium oscillations too. That sounded interesting.
Yeah, really cool. Models show how some cells might encode the strength of an incoming signal, not just in the amount of calcium increase, but in the frequency of these calcium spikes or oscillations.
Wow. Encoding information in rhythm.
Exactly. And then there are gene regulatory networks, understanding the logic of how genes are switched on and off by proteins called transcription factors.
The source mentions some synthetic examples, things designed by scientists.
Yes, like the genetic toggle switch and the repressilator. These are synthetic circuits built inside cells, often bacteria or yeast.
And why are they important?
Because they were actually designed using mathematical modeling principles first. And then they were built, and they show these really surprising behaviors that the math predicted. It's a powerful proof of concept.
Okay, so what kinds of behaviors can these models reveal? The source mentioned steady state behavior.
Right. That's basically where the system settles down into a stable operating point and stays there, like a constant level of some protein.
Or transient behavior.
Yeah, that's the journey to the steady state. How does the system react initially when you perturb it? What path does it take over time before it settles down?
And then the really non-intuitive stuff, like sustained oscillations you mentioned.
Where quantities don't settle down, but instead go up and down rhythmically, forever, ideally. The repressilator is a famous synthetic example. 3 genes shutting each other off in a cycle, leading to oscillations.
And the other really fascinating one was bistability.
Ah, yes, bistability. This is incredibly important. It means a system can exist stably in one of 2 distinct states. Think of it like the light switch. It can be firmly on or firmly off.
And it stays in that state unless you give it a good push.
Exactly. It provides a kind of cellular memory. The system remembers which state it was last pushed into.
And the source said this requires specific ingredients.
Yes, the source highlights it. To get bistability, you generally need 2 key things. Some form of positive feedback in the network, and non-linearity, meaning the responses aren't just simple proportional changes. Things have thresholds or saturate.
And the genetic toggle switch is the classic example.
It's the perfect illustration. 2 genes mutually repressing each other, that positive feedback loop, plus nonlinearity gives you 2 stable states. Either gene A is high and gene B is low, or vice versa.
That's just incredible. The math reveals that these biological circuits can have memory or act like clocks just based on how they're wired and the quantitative details.
It really is.
Yep.
And the sources also touch briefly on the tools needed to actually analyze these complex models, because often you can't just solve the equations neatly on paper.
Right, to complicate it.
So you need ways to visualize what the system could do. Things like phase portraits, which are like maps showing all the possible trajectories the system could take from different starting points.
Okay.
You need methods to find where the stable states are, the steady states, or oscillations that might involve finding nullines where certain rates of change are 0.
And figuring out which parts are most important.
Right, sensitivity analysis.
Yeah.
How sensitive is the model's behavior, say, the oscillation period, or the threshold for switching states to small changes in the different parameters like reaction rates or binding strengths? It tells you where the critical control points might be.
So mathematical tools to analyze the model itself. And because, as you said, these models are often too complex for exact pencil and paper solutions.
You simulate them using computers.
Which brings us back to the beginning almost,
Yep.
to numerical methods.
Exactly. This loops back beautifully. Remember approximating integrals using rectangles or trapezoids from the first source. Well, for complex dynamic models in biology, you use numerical integration algorithms and other computational techniques to simulate how the system changes step by step over time.
So the computer calculates the changes over tiny time increments.
Precisely. The source even mentions methods for dealing with stochasticity, the inherent randomness in biological processes. especially when molecule numbers are small, things like the Gillespie algorithm.
Wow. So it truly comes full circle. The ancient Greek idea of breaking down a curve into infinite pieces connects conceptually to modern calculus tools like integration and differentiation, which in turn provide the quantitative language for building these dynamic mathematical models.
That let us simulate and understand the often non-intuitive complex behaviors hiding within biological networks, from enzyme kinetics to gene circuits.
These models really provide a rigorous way to test our understanding of life, make predictions we wouldn't otherwise think of, and gain insights that just, describing the parts qualitatively simply can't offer.
It's a really powerful synthesis.
It's quite a journey, isn't it? From Greek geometry all the way to understanding oscillating genes and cellular memory switches.
It really shows just how far seemingly abstract mathematical ideas can take us in understanding the concrete reality of life.
Absolutely. So I guess the big picture for you listening is, next time you encounter calculus, maybe don't just see abstract symbols. Remember that its fundamental concepts, rates of change, accumulation, are actively being used right now to decode the most intricate processes of life itself.
And it leaves us with a fascinating thought to ponder, doesn't it? Given the profound ability of these mathematical models to reveal surprising, non-intuitive behaviors in biological systems, things like myostability, oscillations, complex signal processing.
Yeah.
What other fundamental biological processes, maybe ones we currently understand mostly through qualitative descriptions, might be harboring hidden dynamic complexities, non-intuitive behaviors, just waiting to be illuminated by the right quantitative analysis? What else is waiting to be discovered with these tools?
That's definitely something to think about. A great place to leave it. Thanks for diving deep with us into these fascinating sources today.